Hello, everyone. Okay, we are in Unit 9 for AP Chemistry, and we are about to start 9.7, where we're going to talk about the galvanic and the electrolytic cells. And there's a couple of things that I want to do first because I feel like it's a little bit of necessary background information before we start with 9.7. So first things first, let me come over here for just a second and give you a quick overview. In class, I gave you two pictures of this cell, so you can write on them. And I also gave you a Venn diagram, which we'll be filling out the Venn diagram later, not in this video. But what I want you to understand is that there are two different types of electrochemical cells. Chemical cells, there we go. One of them is galvanic. It's also known as there we go. Also known as voltaic. So this is one type, and then the other type is one called electrolytic. Now, excuse me, an electrochemistry part of the AP exam, they spend a lot more time on the galvanic cell than the electrolytic cell, but they do have certain things about the electrolytic cell that they do like, and I will definitely work through some stuff with that with you. But I want to just talk about the galvanic cell first, and there's a lot to it, but it's actually a lot of fun to put it together because it's kind of like a puzzle. Now, on the AP exam, you will definitely have some electrochemical cells somewhere because there will be information about Unit 9, right? But most of the time if you're going to get a big question, it's going to be about a galvanic or a voltaic cell because there's a lot more that we can do with it based upon the information that we learn. Okay, so first thing we're going to do is, this is your blank one. This is why I've given you two. I'm going to go through and number a couple of things on the diagram. Sorry, guys. On the diagram. And then over here on the right-hand side, we'll identify what the parts are. So this will serve as your skeletal structure of sort of what are the parts and pieces of a galvanic cell, what does it look like. That way, when we're building them, you can refer back to the skeletal structure and know exactly what the heck is going on. Okay, so first thing I will number are these two things that look like beakers, which in this case they are beakers. I'll call those number one. Number two is going to refer to these large rectangular things sticking out of the beakers. Number three, I am going to talk about the solution, this liquid that's in the beaker. Number four, I'm going to talk about this thing that's connecting, sorry guys, that's connecting these two rectangular things. This will be number 5, and this will be number 6. Okay, so let's identify these parts because these are all really important when you're drawing a galvanic cell to know what they are, who they are, and so on and so forth, and what they do. I love putting chemical cells together. I think it's a lot of fun, but you got to know what you're working with. So number one, these are just the containers, right? They're nothing special. In this case, they are beakers. There are specialized containers for galvanic cells. That's not something you're going to find in the lab, especially in a high school lab, unless you are in a lab that specifically does a lot of electrochemical cells. They might have them, but usually not. All right, so these are containers. In this case, they are beakers, but they house the two halves of a galvanic cell. So the two halves are called the anode and the cathode. And notice that there are two containers. That's what makes this a galvanic cell. So there's two separate containers. There's the anode and the cathode. I am so sorry. So if you guys think back to Unit 4, when I taught you how to do redox half reactions, so half reactions, so everything is cut in half. So you have the reduction and you have the oxidation half reactions, and they go together to make a chemical reaction. So in electrochemistry, everything is split into the halves. All right, so we have a galvanic cell. We have two containers. All right, number 2, which was up here, referring to these. Okay, so let me get rid of that. I'll have this. There we go. So number 2, these are called the electrodes. And these are part of the anode and or the cathode, depending on which one they are. You will have one anode and you will have one cathode. You can't have two of the same thing. And you can't have just one in a galvanic cell. You have to have one anode and one cathode. So these are your electrodes. They are also the anode or the cathode. Now what you need to know about them is that these are your solid chunk of metal. Okay, it is a solid piece of metal. It is copper, it is silver, it is gold, it is nickel, it is platinum. Well, let's not talk about platinum. We'll save that one for later. Platinum is a solid chunk of metal, but I'll talk about what's special about that one in a minute. But it is a solid chunk of metal. So anything that's a metal, that's a solid piece, that is an electrode. All right, number 3, which I will do here. And 3, just so you can see it, is the solution. Now, this is your solution that goes with the anode and the cathode, right? So depending on which side you're talking about. And in the solution, you should have 1 molar. This is a 1 molar solution that is, I want to go up here actually real quick and do this. Standard conditions. 1 molar or 1 atm for gases and 25 degrees Celsius. Now you will notice that this is pretty much the same all throughout AP Chemistry. The only difference is when you get into the gas laws unit, that temperature changes to 0 degrees Celsius because it's really hard to work with gases at 25 degrees Celsius. But everywhere else in chemistry, it doesn't matter if it's thermochemistry or electrochemistry or whatever, 1 molar, 1 atmosphere, 25 degrees Celsius, which is normal, what they call laboratory conditions. All right, so back to our solutions. This is 1 molar, and these are your ions. Remember, your ions are floating around in solution. So that is really important that you kind of start thinking about this because you're going to have half reactions. And a half reaction will look something like this. It'll be a Cu plus an electron gives you Cu, and then that's a solid. Or you could have Cu2 plus 2 electrons gives you Cu solid. Or you could have Cu2 plus 1 electron gives you Cu plus 1. So these are 3 half reactions, and there'll be some cell potential, some voltage that is associated with these reactions. But what I want you to understand is, let me highlight this one, that ions are what are in solution. So the things that would be in solution would be this ion, this ion, this ion, and this one. And the solid chunks of metal, the electrodes, are going to be any solid chunk that you have. So sometimes, and I'm spending a little bit of time on this because sometimes students get this confused, especially when they're in the situation like this one where there isn't a solid chunk of metal. Ions cannot be a solid chunk of metal. Ions are free in solution floating around. So they are not an electrode. So it has to be a solid chunk of metal in order to be an electrode. And what do you do in this scenario? I'll talk about that in just a second, okay? In the scenario number 3 with the ion to ion. I will discuss that in just a sec. All right, so that's parts 1, 2, and 3. So let's go back to part number 4, which is up here at the top. And it's referring to this here. All right, number 4. So number 4 is the wire. So it's just a metal wire that connects the electrodes. And it happens to go through number 5, which we'll talk about what that is in a second. Number 5 is technically optional. You don't have to have number 5. It'll still work the same way with or without number 5. So I'll go ahead and put that this is optional, but 99.9% of the time you're going to have it. If you have to sketch a cell from blank and they give you nothing, then you make sure you draw number 5, which we'll talk about in a sec. If they give you one that doesn't have whatever number 5 is, then don't worry about it. Just draw the rest of the cell because it's technically not necessary for the cell to operate, but I'll explain. But usually they do put it on there. All right, so number 1, the wire connects the electrodes. And this is what's super major important here. Electrons flow through the wire. Exclamation point. They ask you about electron flow all the time on the exam, and kids get it wrong. They get it wrong because they talk about electrons will flow through what I have numbered as number 6 on this chart. And that's wrong. Electrons flow through the wire. They flow from the solid chunk of metal, one of the chunks of metal, and I'll tell you how to tell which one, through the wire to the other chunk of metal. Think about wire. Wire conducts electricity. Electricity flows. What is electricity? Electrons. Electrons flow through a metal wire. So it's really, 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 really important that you get that and not get that mixed up with anything else. Okay, let's take a look at I'm not going to highlight this one. We'll talk about number 5. This is the one I said was optional. This is your voltmeter. You don't need a voltmeter for the cell to run. But what it does is it will tell you the cell potential. So it will read the cell potential. Cell potential is the capital E. Okay, so that's how much voltage either has to come in or go in or out of the electrochemical half reaction. So, and when you sum them up, the halves, the anode and the cathode, you will get the total, and the total should read on the voltmeter. And we'll talk about that a little bit more because we're going to talk about how we understand this with our batteries and with our cell phones and how they work. And I think it'll help you understand it. But we'll get into that when we get into the actual lesson, not our pre-lesson. All right, number 6, which I'm going to do in green, is this one. Okay, 6 is really important. They're all really important, but this one's called the salt bridge. Okay, think salt in chemistry is another word for what? Chemists call something a salt. What do they call a salt? Think. I'll pause. Okay, go. It's an ionic compound. So hopefully you knew that and you were able to now say that to yourself really quickly. It's an ionic compound. That is what a salt is. A salt bridge. What do bridges do? They connect things, right? So it's connecting the anode and the cathode in this particular, in all the cells that they're in. Now, what it has in the salt bridge is a soluble salt. So think about your solubility rules. You're going to want to pick something that's like a 1A metal with probably nitrate would be one of your best ones. So examples would be like NaNO3, great one, super soluble. KNO3, also super soluble. Know your solubility rules. You want to pick one, just pick one that you want to use all the time and just use it And don't try and like be fancy and switch it up to something different. Just pick one that you know. One of these two is perfect. These are great ions. They're 100% soluble all the time, and there's no question about it. So what they do, what the salt bridge does, is it has the ion flow. Careful. You can see why now maybe kids get this mixed up. Ion flow, because up here we have electron flow. Here we have ion flow. All right, so careful. So it has ion flow from the anode to the cathode, and what it'll do is, get this color. What it will do is it will balance the charges in the cell. So what's happening is you have in this container here, you have some ions from the solution. So that's like what's here. So let's say it's one of the coppers. And then you have over here another ions floating in the solution. So there's some charge in this container. And then as electrons are leaving one of these electrodes, they're going to go from one direction to the other, and we'll discuss that. But as electrons are leaving, things are happening. And your ion concentration that's here in the blue is either going to be increasing or decreasing. And when that happens, that causes your cell to stop because you want to maintain the balance of your charges. So in order for us to maintain the balance of the charges, that's where the salt bridge comes in. Because the salt bridge, you can put in a soluble salt, and we know that these soluble salts don't do anything. They are not going to react with anything else because they're always soluble in the solution. They're just going to go to one beaker or the other, and they're going to do that based upon where their charge is needed. Be it, yellow is probably not best, be it that it splits into an Na+, which is a positive charge. So if you lose your positive charges for some reason, you'll add your Na+ over there, and the NO3 will go to the other side. So that's your negative. So it depends on what charge you need to replenish is how it will split itself up, and they'll go in opposite directions. Okay, so these are the massive parts. So let's just take a second. Let's look back at what we've got. I have galvanic cell, which has two containers. I have the anode and I have the cathode, and I don't know which one's which, and it doesn't matter, and I'll talk about that a little more. But the entire anode or cathode includes the entire thing. Okay, so that's the entire anode and cathode. Now, inside each one of these, I have an electrode, check, check, and it's a solid chunk of metal. And then I have a solution, check, check. And it should be 1 molar. Now, it could be a different molarity, but it's not going to be standard conditions. And in each one of these, I have one half reaction. Now, I'm not going to have a copper more than likely, but I would have copper with silver or something like that. So I have my solutions. I have the wire, which the electrons flow through. And that's super important. The voltmeter reads the voltage, so the cell potential. And then I have the salt bridge, and the purpose of the salt bridge is to let let the cell run longer, okay? So it's soluble salts, and its purpose, so we'll go back down here and add, its purpose is to allow the cell to run longer. Okay, so the cell can run longer. If you take the salt bridge out, the cell is going to very quickly run out. And basically what that means is the voltage will run to 0. So you'll see the voltage just drop to 0, and then it's done. It's going to not run for very long at all if you don't have the salt bridge in there. And with the salt bridge, it will not run indefinitely. It's just going to run longer than it would without it. So that's why we have the salt bridge there. All right, so some of the mnemonic devices that we've learned in the past were oil rig. Remember, oxidation is the loss of electrons and reduction is the gain. of electrons. Remember? All right, so that's oil rig. And we have anox and red cat. And anox is oxidation, is at the anode. Red cat is reduction, is at the cathode. All right, so we have our two cells. There's an oxidation half and there's a reduction half. The oxidation happens at what we call the anode. The reduction happens at what we call the cathode. And then let's talk about that before I give you the last one. Get some of the writing off because it starts to get a little crazy. All right, so here's the deal. Does it matter which one is the anode and which one is the cathode? In terms of when you're starting with a blank slate, no. It doesn't matter where you put it, meaning left or right. Most textbooks and most sample problems are going to put the anode on the left and the cathode on the right. And it's just something that people do. However, as we have learned, AP picks up on little tricks when people are trying to take shortcuts. And so the next pneumatic device I'm going to give you talks about the flow of electrons. And let's go ahead and do that and then I'll come back. The flow of electrons comes from our next pneumatic device, which is FAT, CAT. And what that means is electrons flow from anode to cathode. So that's where fat cat comes from. So from anode to cathode. So if the standard is to put the anode on the left and the cathode on the right, then that means the electrons are going to be flowing from the left to the right. Well, on the test, many students started saying, oh, well, when they asked them which way the electrons flow, they go, oh, it goes from left to right, always, because every sample problem they ever worked, the electrons always went left to right. And then they would mark it wrong, and so then AP started flipping it. So they would put the cathode on the left and the anode on the right. So don't make any assumptions of which one is which. You have to figure out who is the anode and who is the cathode based upon who's being oxidized and who's being reduced. So this down here, that determines who's the anode and the cathode. And if they already have the half reactions written out on the cell for you, then you just go with that and identify who is the anode and who is the cathode. If it's blank, like this one, then you pick. It doesn't matter. You can put the anode on the left or on the right. It's irrelevant. Because let's face it, if you had this sitting out on a table, if you look at it from one side of the table, the anode could be on the left, on the right, and you walk around the table, and then it flips. So it doesn't matter. Whatever you want to do. So just pay attention to what they have written out for you, but stay away from notions of, oh, electrons flow from left to right. That's a wrong answer, and they will not give you credit for it, even if the anode is on the left. You have to say the electrons flow from the anode to the cathode. That's it. You got it? Okay. All right, so this is your basic information on a standard cell potential of the bits and parts of it, because coming up I'll make another video where we will actually build a cell and I'll show you how to do that and how to make that happen. I just really want to emphasize that your electrodes are a solid chunk of metal. They are not ions. And your solutions are 1 molar. Oh, and I will go ahead and address this now, might as well. So what do you do here when you have either an ion to an ion issue or the half reaction doesn't have a metal in it? All right, so if the half reaction has non-metals, because we have some of those too. So you can split water. You can have half reactions for water. You can have half reactions for like bromine and things like that, which aren't metals. So how do you have a solid chunk of metal? So when you have ion to ion situation, like is shown here with this copper, the third copper, or you have a non-metal half reaction, then you have to use what we call an inert electrode. Okay, the word inert means non-reacting. It doesn't react, it's inert. So an inert electrode would be platinum. That's why I said that was not the best thing to say for a solid chunk of metal, but it is a solid chunk of metal. You can use platinum, and actually another one that you can use, and you're like, but wait, that's not metal. I know, but it still works, is graphite. And graphite is a form of carbon. Actually, when I would run labs with this, we would pull out pencil leads from our mechanical pencils and we could use those as our electrodes if we needed an inert electrode. So platinum or graphite will work as your inert electrode. Just FYI, most people, when they're writing it, they would identify it as platinum because it's just easier to write PT than all the, writing out graphite, because we're trying to save time when we're working, so we go as quick as we can. But many times on the AP exam, they're just going to write out graphite. So just be aware, you use an inert electrode in the sense when you have ion to ion, like in this third copper one, or if you have nonmetals. Okay, that's enough for galvanic cells. Absorb that. That seems like a lot, and it seems a little overwhelming. I will make another video where we build a cell, and it's going to look just like this one, and we'll put it all together, and you'll see, and it kind of hopefully will make more sense. Okay, guys, bye.